So this is our second topic for this week and the first topic of endocrine, the other one being thyroid disorder. So in this one we're going to talk about diabetes mellitus, and of course we can divide that into type 1 and type 2. We're going to follow the same format, so we're going to talk about the underpinning pathophysiology first. So this is the pathogenesis. This is what's happening to cause the actual disease itself. In the second part of this video, we'll look at the applied pathophysiology. So we'll look at the most common signs and symptoms and how that underpinning pathophysiology then causes those signs and symptoms, if we know at all. We'll also talk about diabetic complications because, of course, this is a big issue in terms of diabetes. So, without further ado, let's start with type 1. Type 1 is primarily genetic, but there's also thought to be an influence from environmental factors. There is some evidence that gene expression becomes altered after infection with specific viruses, and so far they've identified rubella and a few others that have been implicated in triggering type 1 DM. The other thing with viruses is that they induce pancreatic beta cell apoptosis. This, of course, would be a normal immune response to that virus. The problem with this is that our antigen presenting cells potentially then take normal host proteins back and stimulate an immune response to those normal or slightly altered host proteins. Once that happens, of course, you'll remember adaptive immunology develops a memory. So it develops a memory, so it continues to produce both T and B cells against those host proteins, and they will go out and they will treat those proteins, those normal host proteins in the pancreas, as antigens and will attack them. So type 1A is classified as a type 4 delayed type hypersensitivity. Now, these typings are part of a classification by a couple of guys called Jellincoombs. And although there have been attempts to use other classifications and adopt other classifications, because it's quite old now, nothing else really has managed to classify it as clearly. So any hypersensitivity disorder tends to be classified by this Jellincoombs classification system. And according to that, this is a type 4 delayed type hypersensitivity. This is an abnormal immune response, and it's the same hypersensitivity response that you get with Hashimoto's thyroiditis, which we'll talk about in the next video. What happens is you produce activated antigen-specific, remember this is the adaptive immune system, T lymphocytes, so T cells, and activated antigen-specific B lymphocytes, B cells. B cells, of course, once they're activated, then become plasma cells. They differentiate into plasma cells. Both of them produce memory cells so they can keep on attacking. Once B cells become plasma cells, they secrete antibodies. In this case, they are autoantibodies because they're attacking our own proteins. And they are attacking specific proteins because, of course, the adaptive immune response is antigen specific, and those autoantibodies, if you remember the structure of antibodies, at the bottom we have the Fc, the fragment constant. At the top we have the fragment variable sites, that top of that Y shape in a monomer. The fragment variable sites are what are specific and what bind to these antigens. So our specific activated cytotoxic T lymphocytes, remember TC cells, destroy pancreatic beta cells. If you remember, what they do is they use their T cell receptor to bind to the target antigen, and then they secrete perforins and granzymes to cause apoptosis. So potentially they could cause apoptosis of the pancreatic beta cells that of course synthesize and release insulin. Pancreatic beta cells can also be opsonized by the autoantibodies. Remember, this is where antibodies bind to the target via the Fv, and the Fc can then bind to phagocytes, or in antibody-dependent cellular cytotoxicity, ADCC, the Fc can bind to natural killer cells. Natural killer cells then, like cytotoxic T lymphocytes, secrete perforins and granzymes, causing apoptosis. Now, beta cells, of course, they are epithelial cells. If you remember, there are two types of epithelial cells. We have our covering and lining type, and we have glandular. So these are epithelial cells, and they have the common characteristic of epithelial cells in that they can proliferate quite quickly. They can regenerate. But if the destruction is too great, they can't regenerate fast enough. So our common type 1 that we generally talk about is called type 1A. This is the type for hypersensitivity. Type 1B does exist, but it is rare and mainly found in Asia and Africa. It's not an autoimmune response, but it does involve an absence of insulin. Okay, so they can't regenerate fast enough, the beta cells. So once many of them have been destroyed, insulin secretion is going to be limited, synthesis and secretion. Therefore, of course, glucose uptake is going to be low, and if it's not taken into the cells, it's going to remain in the blood, and hypoglycemia occurs. So why is glucose uptake low? You know this, you should know this from physiology. If you remember, insulin binds to tyrosine kinase receptors. Remember, those are those two receptors that come together and they form these dimers of tyrosine, and they then self-phosphorylate. That's the signaling mechanism that in most cells will upregulate GLUT expression. GLUT, of course, being the glucose transporters. Some cells that we'll talk about later aren't dependent on insulin to do this, but most cells are. The other complication here is, remember pancreatic alpha cells. Pancreatic alpha cells synthesize and secrete glucagon. They also have these tyrosine kinase insulin receptors. So what happens, their GLUT1, when insulin binds to these receptors, are expressed. Of course, if you increase the GLUT in an alpha cell, more glucose is going to get into the alpha cell. And because of the way these alpha cells work. If you can't remember, it was in your pre-reading for this section. If glucose can't get into the alpha cells, then it inhibits glucagon release. But in this case, the insulin is not being released, therefore it cannot bind to the alpha cells, therefore the glut cannot be expressed. The alpha cell's mechanism, of course, is going to have low ATP. This low ATP is going to, through those OTP-sensitive potassium channels, stimulate glucagon release by exocytosis. So the alpha cells think that the body is in a low glucose state because they're missing the glut. And consequently, they release glucagon. Glucagon, of course, then goes off and breaks down glycogen that's stored in the liver, and this will then increase hyperglycemia. So there is a paradoxically high level of glucagon, even though there is hyperglycemia, and that then increases the hyperglycemia by breaking down glycogen from the liver into glucose. So glucagon stimulates further increase in hyperglycemia by glycogenolysis, that's the release of the glycogen and breakdown of those big chain sugars into glucose, and glyconeogenesis. Glyconeogenesis is the synthesis of glucose from amino acids. In addition, if glucagon is strongly stimulated, if you have lots of glucagon exocytosed, it stimulates lipolysis, the breakdown of fats for energy. So let's have a look at that in the form of a diagram. Remember, this is type 1 diabetes mellitus. So in comes a load of glucose. That load of glucose goes round as blood circulation and enters the beta cell. Unfortunately, in this case, of course, the beta cells are either dead or dysfunctional, so it doesn't stimulate the production of insulin. Because there's no insulin, there's going to be no increased glut expression in most of the cells, in most of the tissues throughout the body, so that glucose that's in the circulation cannot be taken up by those cells. Paradoxically, of course, this also means that there's no glut expression in the alpha cell. The alpha cell thinks that blood glucose is low because none is getting into it, so it exocytoses glucagon, which it would normally do in fasting conditions. That glucagon goes off to the liver. Here we get glycogenolysis, so glycogen is stored in the liver, great long chain molecules of carbohydrate are converted back to glucose. This is then released into the circulation, which increases hyperglycemia even more. Which brings us on to type 2 diabetes mellitus. Again, we have a genetic component, so there is often a family history. There is often a strong effect from environmental factors, such as overeating, obesity, inactivity, smoking, alcohol, stress, and aging, all those things that we shouldn't really do. Not that we have a huge control over aging, but maybe some of the others. So In addition to the inherited genetic abnormalities, lifestyle factors can then also cause genetic polymorphisms. They may change gene expression. So alterations to the genes or their related sort of DNA-based transcription receptors. Remember, protein synthesis always begins with a transcription factor binding to a receptor on DNA. Any changes along there is going to affect the insulin receptor or any other component of the signaling pathway. Remember, we don't always talk about the complete complex signaling pathways. So if you have an issue with any component of that signaling pathway, it's going to cause a problem. This leads to insulin resistance. So although beta cells are releasing insulin, insulin cannot stimulate the upregulation of GLUT because of issues in the signaling pathway. And again, we have this issue, we have the same effect. If insulin cannot stimulate the upregulation of GLUT, it also can't do it in the alpha cells. So again, we're going to get a paradoxical exocytosis of glucagon from the alpha cells, which is going to break down our glycogen, which is going to increase hyperglycemia further. What can happen eventually, and this is why it's important in type 2 diabetes to try and modify people's lifestyle factors, because if insulin resistance continues or worsens, it can eventually impair insulin secretion, because what you're doing is you're constantly stimulating an excess of insulin in an attempt to try and reduce the hyperglycemia. And ultimately, this will stress the beta cells and they will start functioning poorly and not actually releasing insulin. So type 2 can start to develop into more of a type 1 condition. And this is often where people with type 2 start needing to have insulin. Let's have a look at another diagram for type 2. So, okay, in this scenario, again, we've got high levels of glucose in the blood. We've got hyperglycemia. Glucose is taken in, it gets into the beta cell. The high intracellular glucose stimulates the exocytosis of insulin in this case. Remember, in type 1 we didn't get insulin. In this case we do get insulin. That goes off to most cells throughout the body. But it still results in no additional expression of GLUT transporters because of issues in the signaling process. Insulin resistance within those cells. So GLUT are not increased, that glucose can't be taken up by those cells, so it remains in the blood, causing hyperglycemia. In addition, because of these issues, the insulin cannot signal alpha cells to express GLUT, so the alpha cells don't express GLUT. This is interpreted as hypoglycemia because it is within the cell rather than hyperglycemia, and the alpha cells exocytose glucagon. Glucagon, of course, glycogenolysis, also known as glucogenolysis in some texts, glycogen is converted back to glucose, which then is released from the liver, from the hepatocytes, and increases that hyperglycemia further. Well, that brings us on to applied pathophysiology. So here we start talking about the common signs and symptoms and also the complications. So we'll talk about polyuria, glycosuria, polydipsia, polyphagia, and weight loss, what I often refer to as the three polys. Also blurred vision, neurovascular complications, and we'll talk briefly about ketoacidosis. So first up, glycosuria. So, okay, if excessive glucose is lost through glomerular filtration, bear in mind during filtration, everything is filtered out. Things that we want, we then put in mechanisms in order to pull them back from the renal tubules into the circulation. Now, normally glucose is reabsorbed by SGLTs in the renal tubules. However, if lots of glucose is filtered out into the tubules, those SGLTs become overwhelmed. So the glucose ends up remaining in the tubules to be excreted in the urine, causing glycosuria. This leads us on to polyuria. Because glucose is an osmotic molecule, this is going to change the osmotic gradient. If there's an excess of it in the filtrate in the tubules, then it's going to change the osmotic gradient. It's going to retain fluid within the tubules and prevent its reabsorption through aquaporins back into the circulation. So water isn't reabsorbed, it's excreted, so we get this osmotic diuresis. Polydipsia. Of course, if we're losing a lot of water via the urine, it's going to stimulate hypovolemia, and we know hypovolemia stimulates RAS. We've also got a high osmolarity in the blood because of the glucose there, so our osmoreceptor is going to shrink. Because of this, we're going to stimulate the secretion of ADH, antidiuretic hormone. Antidiuretic hormone also stimulates thirst. So as we said, the high glucose content in the blood causes hypertonicity. This then stimulates those hypothalamic osmoreceptors to shrink, which also promotes thirst. Polyphagia, eating lots. I think possibly we all suffer from slightly too much of that, but not, unfortunately, with the weight loss. In this case, we've got polyphagia and weight loss, and this is mainly in type 1 DM. So, of course, there's going to be cellular starvation of glucose because the gluts aren't upregulated in either type. So, we can't get glucose into the cells. Because we can't get glucose into the cells, we can't use that glucose to make ATP. So we're going to turn to alternative sources of energy. These alternative sources of energy, carbohydrates, lipids, and proteins, are all going to be broken down for energy because we can't use the glucose. When they're broken down, of course, then we start to get weight loss. We're also, because of this cellular starvation through the various pathways that stimulate hunger, we're going to get polyphagia, this constant sensation of hunger. Also contributing to the weight loss, of course, is the hypovolemia, the water loss through that osmotic diuresis. This is what triggers hunger, but also promotes weight loss. Blurred vision. is due to something called the polyol pathway, not to be confused with polydipsia, polyuria, polyphagia. This is a metabolic pathway that leads to the production of sorbitol within cells. When we talk about sorbitol, yes, it is the same thing that you get in sugar-free mints and sugar-free chewing gum. So what happens is you end up getting a sorbitol accumulation in the epithelial cells of the lens and the retina, which contributes to blurred vision. As we said, intracellular sorbitol is created via the polyol pathway. So let's look at that pathway. Several cells, we talked about this before, are non-insulin dependent. So in other words, they don't need insulin to take up glucose. This includes retinal cells, renal endothelium, the epithelium of the lens, Schwann cells. Remember, Schwann cells myelinate neurons in the peripheral nervous system, and also neurons themselves. All of these cells can transport glucose intracellularly without insulin. They already have enough glut to be able to do this without requiring insulin. And of course, you can see what's going to happen here. If we've got hyperglycemia and we've got these cells that already have some gluts, they don't need insulin, they are going to absorb a lot of that excess glucose from the blood. So when high levels of glucose enter these cells during hyperglycemia, the excess is converted to sorbitol. Now this is a normal physiological process, and it gives us a store of sorbitol that can be then converted back to fructose, which can then be used for cellular respiration. But, and this is the common problem, as you saw before with SGLTs, if we don't have enough of the structures or enough of the enzymes required to deal with excesses, those excesses, the pathways become saturated due to the excess. So if the pathways that convert sorbitol into fructose are saturated, so we've already converted our glucose into sorbitol, but now we can't convert it into fructose, sorbitol is not converted. Sorbitol cannot get out of the cell either. So sorbitol can't cross the plasma membrane, so it accumulates intracellularly. And this, of course, sorbitol is, as you probably know, if you eat too many sugar-free mints with sorbitol in or chewing gum or whatever, you get diarrhea. This is because sorbitol is an osmotic molecule, so it will draw water to itself. In terms of the gut and diarrhea, of course, the sorbitol is drawing water into the lumen of the gut, which gives you diarrhea. In this case, what happens is water wants to influx, it wants to move in to these cells that accumulate sorbitol. This is hypotonicity because here we have an osmotic gradient that means water wants to move into cells. Another problem with this is there is an increased risk of oxidative stress because the conversion of excess glucose to sorbitol has a bit of a knock-on effect in that it reduces one of the key intracellular antioxidants. Now, if it reduces this antioxidant, it increases the risk of oxidative stress within the cell because you are lacking the antioxidant to deal with any oxidation. So, in relation to the eye, we have effects on the lens. So sorbitol collects in the cuboidal epithelial cells of the lens. This creates an osmotic gradient, as we explained, resulting in swelling of those cells and swelling of the lens due to this water influx. And obviously this is going to have an impact on refraction, because we need to be able to change the shape of that lens to enable the light to focus on the retina. In this case, refraction is changed. In the longer term, it is theorized that this sorbitol accumulation, and this seems quite logical, contributes to the development of diabetic cataracts. In terms of the retina, as we said, retinal cells can also transport glucose in independently of insulin, so therefore they are also prone to sorbitol accumulation. This causes the diabetic retinopathy because we have dysfunction of those retinal cells because of this sorbitol accumulation, water influx. And this gives reduced vision or blurred vision. Of course, this is then exacerbated because in diabetic retinopathy, there will, because of these dysfunctional cells, potential death of cells, be inflammation. There will also potentially be damaged neurons. And remember, we said neurons can also move glucose in independently of insulin. So they are also prone to sorbitol accumulation. And it's these damaged neurons that give those cotton wool spots. Because of inflammation, we're going to have increased vascular permeability. And there is also, because of inflammation and various growth factors being released and various other mechanisms, neovascularization, so new vessels grow. And this gives that common look of the retina in uncontrolled diabetes. Some of these changes are caused by something that we're going to talk about in a minute. These are advanced glycation end products, aka AGEs, AGEs, which cause oxidative stress and inflammation. There is also, again, we'll talk about this in a minute, upregulation of DAG. Remember DAG? DAG, diaglycerol, was produced when our phospholipase PEP complex was activated by an alpha subunit. DAG was one of the things produced, the other one was IP3, which caused the release of intracellular calcium. DAG, of course, went off and activated protein kinase C. So there's an upregulation of DAG, which causes an upregulation of PKC, protein kinase C. There's also an upregulation of vascular endothelial growth factor, VEGF. And this contributes, of course, to the neovascularization. Neurovascular complications involves a complex interaction of 4 abnormal or adapted pathways in hyperglycemia. You can see there we've got the polyol pathway that we've already talked about. In the polyol pathway, as we said, there is sorbitol accumulation inside cells that can take up glucose independently of insulin. There is also a reduction in a key antioxidant. AGEs, advanced glycation end products, when they're created, they have effects, quite widespread effects, on protein modification. They also stimulate the production of ROS, reactive oxygen species. This is a free radical that is often involved or increased in inflammation and inflammation itself. Protein kinase C, of course, we said, is upregulated because DAG is upregulated. This leads to vasoconstriction, angiogenesis. Angiogenesis is the same as neovascularization. We have new vessels growing, budding off from existing capillaries. Generally, this supports healing tissue, but it can also happen in pathological conditions and cause additional problems. Increased vascular permeability. So if we have increased vascular permeability, of course, we're going to have transudate and potentially exudate moving out of the circulation into the tissues. There is the reduced breakdown of fibrin. So fibrinolysis, lysis being breakdown, is the breakdown of fibrin. And this is reduced. So we're going to have the potential for fibrosis, for scar tissue developing. Our other one, N-acetylglucosamine, lovely word that for you, one for your glossary. This has an effect on altered gene transcription. And you can imagine that again is potentially has quite devastating effects on the functions of cells. It also increases TGF-beta, transforming growth factor beta. So it increases one of the key growth factors. It also increases something called PAI1 that we'll talk about in a minute. All of these, as I said, there's this complex interaction because as a result of ages, we have increased ROS production, that free radical. That then has a knock-on effect on all of those pathways. It upregulates all of those abnormal pathways. So a little bit more detail on the polyol pathway in terms of this antioxidant issue. When glucose is converted to sorbitol intracellularly, it's converted by aldose reductase, which is obviously an enzyme. It uses a cofactor in this conversion, NADPH. You don't need to know what that stands for, but I'm sure you can remember NAD. This is NADPH. NADPH also regenerates the intracellular antioxidant that we were talking about before, which is called glutathione. In some texts, it has a slightly longer name. We're just going to call it glutathione. So, of course, if it's busy being a cofactor in the conversion of glucose to sorbitol, it's not going to be available to regenerate this antioxidant. So, therefore, if NADPH is busy, is consumed by sorbitol production, no spare time, no spare capacity, glutathione is not going to be available to prevent oxidative stress. When this happens in neurons, remember neurons are one of the cells that use the polyol pathway in hyperglycemia because they are able to transport glucose in independently of insulin. In neurons, the oxidative stress reduces action potential conductivity. So in other words, action potentials do not conduct as well or as rapidly as they would normally do. So we have neuropathy. AGEs are advanced glycation end products are created by the process of advanced glycation. Now, glycation is the non-enzymatic, in other words, it doesn't require enzymes, reaction between sugars, which of course includes glucose, and proteins, lipids, or nucleic acids. So this is a reaction that doesn't depend on having the presence of certain enzymes. It happens, if you like, automatically, independently, freely. What this then creates is these advanced glycation end products, these AGEs, when this happens. Now, here comes our dear old friend, reactive oxygen species. Reactive oxygen species comes in, oxidizes the advanced glycation end products, and we get an increase in them because it causes the breakdown of these existing AGEs into numerous other AGEs. In addition to this process, where AGEs are produced endogenously by advanced glycation, by our glycation of proteins and lipids, etc., they can also be consumed. And this is the big issue really with heat processed foods, because AGEs are consumed via heat processed foods, by cooked foods. In patients with diabetes type 2 in particular, AGE formation is endogenously increased. Of course, eating them as well doesn't help. AGE accumulation, it's not just about diabetes though. It's found to be implicated in several other chronic diseases, including frailty in aging, atherosclerosis, so the formation of atherosclerotic plaques leading to cardiovascular disease, Alzheimer's disease, rheumatoid arthritis, and chronic heart failure. So AGEs are not good things when they accumulate. So what do? They modify intracellular gene transcription and some of the signaling molecules. And this is quite complex what they do and quite broad in what they do. So the effects can be quite significant. They also, as we said, modify proteins, particularly extracellular plasma proteins such as albumin. And we have an increase in reactive oxygen species, which of course contributes to the accumulation of AGEs. So the interaction of AGEs with their receptors, sometimes called RAGEs, increases reactive oxygen species secretion. It also activates NF-κB, nuclear factor kappa B. Now, NF-κB is a key intracellular signaling molecule in inflammation. It is the signaling chemical, signaling molecule that is activated when phagocytosis happens or there's any cellular damage, and it immediately causes a cascade of reactions that causes genes to be activated that then produce the cytokines, the chemokines, all those inflammatory mediators that are synthesized in inflammation. So it's a key inflammatory signaling molecule. So ROS causes oxidative stress, NF-κB upregulates inflammatory mediators, leading to, of course, oxidative stress and inflammation. Protein kinase C, this is produced because there's an upregulation in DAG. DAG then stimulates protein kinase C. Increased protein kinase C, if you remember, protein kinases, all they do is phosphorylate. They phosphorylate other enzymes, lots and lots of other enzymes that might have stimulatory effects, that might have inhibitory effects. So we have the potential for lots and lots of different effects if PKC is increased. One of these is vasoconstriction. Now, vasoconstriction in this case is caused because PKC reduces nitric oxide production. It does this by reducing the enzyme eNOS, endothelial nitric oxide synthase, that leads to the production of nitric oxide. Of course, nitric oxide is a key physiological vasodilator. It usually works locally to vasodilate if there's any obstruction or any issues. If its production is reduced, then of course we're not going to have that ability to vasodilate when we need to. So the opposite being vasoconstriction. PKC also increases something called endothelin 1. Endothelin 1 is a vasoconstrictor. So it causes vasoconstriction. So we've lost our vasodilator and we're actively promoting vasoconstriction with PKC. As we said before, PKC also increases transforming growth factor beta, TGF-B. This ends up with an increased extracellular matrix. So we lay down more collagen around the cells. This gives us sclerosis and fibrosis. And this is a major contributor to neuropathy and nephropathy. And here's our old PAI1 that I promised. This is plasminogen activator inhibitor 1. PAI1, you can see why they abbreviate it. And this causes reduced breakdown of fibrin in clots. So we're going to get increased clotting risk. PKC also increases vascular endothelial growth factor, VEGF, and this contributes to the angiogenesis or the neovascularization and the increased vasopermeability. And this is commonly responsible for contributing to retinopathy and also nephropathy. So we said this was about neurovascular complications. So there is an interaction. These two are interdependent, really, in terms of complications. Vascular complications have an effect on neuropathy, on the nerves. All afferent and efferent, so sensory and motor and autonomic neurons are affected by diabetic neuropathy. Autonomic neuropathy, often known as DAN, diabetic autonomic neuropathy, of course is going to lead to organ dysfunction because this is our sort of subconscious, if you like, nervous system, part of the motor nervous system. that regulates the function of organs. The sensory branch of this also monitors what's happening in terms of the body. So autonomic neuropathy is going to lead to organ dysfunction and the symptoms of orthostatic hypotension, urinary symptoms, and GI effects. Diabetic peripheral neuropathy, DPN, this is what gives that bilateral stocking glove pattern of sensory and motor loss. So in other words, the hands and lower arms are affected, the feet and lower legs are affected, stocking and glove. And this really, if you remember, we said conduction speeds are going to be reduced. And conduction speeds are going to be more critical in the longer nerves. So hence it tends to affect these areas first. There is also the potential for damage to motor neurons, and this often leads to deformity and altered gait. And this commonly results in diabetics putting more weight onto the ball of the foot, because you end up getting this very high arch, which puts the pressure on the ball of the foot. And this is why diabetic ulcers will often appear there first. And of course, because of sensory loss, they may not know they're there. So apart from sorbitol damage to nerves, oxidative stress and sclerosis causes reductions in blood circulation to neurons, which causes hypoxic stress in those neurons in addition to the other issues. Autonomic neuropathy, of course, is going to have an impact on vasomotor regulation, which is also going to cause issues in terms of perfusion of the tissues. Another issue is C fibers. Remember C fibers from your pain lectures. They release, when they're stimulated, neurokinins. Now, those neurokinins tend to cause vasodilation around the area where they've been stimulated. So we have calcitonin gene-related peptide, which binds directly and causes the relaxation of smooth muscle cells of the vasculature, causing vasodilation. Or we have substance P, which degranulates mast cells, which of course releases histamine, which is a vasodilator. Now, if diabetic neuropathy affects those C fibers, there's going to be a reduction in the release of these vasodilating neurokinins, contributing to vasoconstriction. That vasoconstriction, of course, is then going to have a knock-on effect on the perfusion to neurons. So there's quite an interaction going on here in terms of the neurovascular complications. So that was our 4 modified pathways that all contribute to neurovascular complications. And you can see not only is there an interaction within those pathways, there's also an interaction in terms of the neuro component and the vascular component of these complications. So that brings us on to ketoacidosis. As you're aware, I'm sure, ketoacidosis occurs mainly in type 1 DM, but it can occur in type 2, particularly if these patients have kept stimulating the release of insulin and they now have impaired secretion of insulin. Ketoacidosis is stimulated by a combination of low insulin and high glucagon levels. Remember, we said there's this paradoxical release of glucagon. Glucagon, of course, stimulates glycogenolysis, so the breakdown of glycogen to provide glucose, and gluconeogenesis or glyconeogenesis. This is the conversion of lactate, glycerol, and amino acids to produce more glucose. Of course, this glucose can then not be taken up by the cells either, so we have cellular starvation and we have increased hyperglycemia. Now, if you remember, we said in high hyperglycemia that's sustained, eventually glucagon will stimulate lipolysis, the breakdown of fats. This releases free fatty acids, not three, although there are three, free fatty acids, and they release them from triglycerides. These fatty acids enter cells and they become oxidized by a process called beta oxidization. This happens in the mitochondrial matrix, and what the upshot is that this process creates acetyl-CoA. And if you remember, acetyl-CoA then goes into the Krebs cycle, and then we have the whole end of cellular respiration, if you like, involving the Krebs cycle and the electron transport chain to produce ATP. Lipolysis is the process where lipases separate triglycerides. Triglycerides, if you remember, are three fatty acid chains with a glycerol molecule on top. The lipases separate these three fatty acid chains, so they create three free fatty acids from each triglyceride. The glycerol, as we suggested above, is then converted via gluconeogenesis using glucagon to produce more glucose. The free fatty acids go into the cells, into the mitochondrial matrix, become oxidized to create acetyl-CoA. Now, insulin would normally inhibit lipolysis. So where insulin isn't being produced, so in type 1 diabetes, there are low levels of insulin. This low level of insulin then allows lipolysis to go unchecked. It allows unregulated lipolysis, so we get repeated breakdown of the triglycerides to release these free fatty acids. So what does all this mean in terms of ketoacidosis? Ketoacidosis starts with ketosis, the creation of ketone bodies. Ketogenesis, the creation of ketone bodies, occurs in the liver. Now, ketone bodies are acetoacetate, beta-hydroxybutyrate, and the byproduct of acetoacetate breakdown, which is acetone. Hepatocytes, they're very generous. When they produce acetyl-CoA from fatty acids, they convert it into ketone bodies rather than using it in their own cellular respiration. When they convert it to ketone bodies, still within the mitochondrial matrix, they release the ketone bodies into the circulation for other cells to use. So hepatocytes release these ketone bodies. The other cells can take the ketone bodies up and then convert the ketone bodies back into acetyl-CoA for use in cellular respiration. Okay, so there's going to be an increase in ketone bodies here, partly because we've got uncontrolled lipolysis. Remember, insulin regulates lipolysis. It reduces the amount of lipolysis going on. If there's no insulin, we're going to have lots of lipolysis, lots of breakdown of triglycerides into glycerol and free fatty acids. In addition, we've also got this action of glucagon in significant hyperglycemia, where they also stimulate lipolysis. So we get, because of the action of the hepatocytes, an increase in the release of ketone bodies because there's an increase in the availability of fatty acids. This ends up with this situation again of saturated metabolic pathways because they end up producing more ketone bodies than the cells can convert. Their metabolic pathways become saturated. Oh yes, it's all very nice, Mr. Liver, you releasing all these ketone bodies for me, but I've got enough, thank you very much. I don't have anything to convert them with anymore. So we have this oversaturation. And of course, you can see what's going to happen. We're going to end up with ketone bodies floating around in the circulation, unwanted. So ketone bodies end up circulating in high concentration, giving us ketosis. Eventually, due to the release of H+ from these ketone bodies, this will give us ketoacidosis. And this occurs when we have a ketosis of something like more than 12 millimoles per litre. In addition, some of the acetoacetate in circulation breaks down, and this releases acetone. And acetone, we don't use acetone, is exhaled, this is where we get the pear drops breath, or excreted in urine. This is where we'll do our dipstick and, oh yes, look, there's some ketones. So going back to moving in from ketosis to ketoacidosis. As I said, ketone bodies, when they're in circulation, lose protons. They lose H+. So we're going to get lots of H+ building up in the circulation, which gives us, of course, metabolic acidosis. Now, normally, any free protons flying around will be buffered with bicarbonate, HCO3- to form carbonic acid, H2CO3. And if you remember, carbonic acid, this is the reverse of our conversion of CO2 to bicarbonate and protons. So this is the reverse, so we're going back to carbon dioxide. Carbonic acid will then dissociate and give us carbon dioxide, which we can get rid of. So normally this buffering process is quite handy. But again, it's like all things. The body can only cope with so much. So if there is an excess of H+ flying around, this buffering process becomes overwhelmed. We don't have enough bicarbonate to buffer it. In addition, of course, this can result in compensatory hyperventilation due to chemoreceptor activation. And going back to where we started with our signs and symptoms, there is also the potential for dehydration because of osmotic diuresis. So we talked before about those SGLTs that reabsorb glucose from the urine back into the circulation. They become overwhelmed, don't reabsorb the glucose, we get glucose in the urine, which creates an osmotic gradient and draws water so that we excrete more water, causing dehydration and hypovolemia. Excreted ketones have an effect here too. When they are excreted, they have to be buffered with sodium, Na+. And of course, this is also going to draw water into the renal tubules, giving us even more osmotic diuresis and causing even more dehydration. The large fluid loss also depletes other ions. So we can get all sorts of electrolyte imbalances going on. Which brings us to a nice diagram as a summary, not one of my own in this case. I nabbed it and I have a link to it there if you want to see it online. This really sums up all of the different processes that are happening in insulin deficiency or in significant insulin insensitivity. And I'm not going to go through it in too much detail. You can use that as reference. And what I'd suggest you do is once you've been through the content a few times, downloaded the PDF version, and maybe make a point of trying to learn it all, have a look at this summary and work your way through the steps, adding the detail in your head as if you're explaining this diagram to someone. And that might be a good way of learning this and retaining it. And I'll see you next in thyroid disorder.